Hello, and welcome to Field Lab Earth, the podcast that's all about past and present advances in the fields of agronomic, crop, soil, and environmental sciences. Today, we'll be talking to Drs. Nidhi Rawat and Vijay Tiwari. Wheat Fusarium head blight, or FHB, is a disease that results in yield losses via mycotoxin-contaminated grain. Finding ways to combat this disease is an important area of research, but long breeding times can delay progress. This episode, Nidhi and Vijay join us to discuss their work testing speed breeding, a technique that may cut current breeding times in half. I'm your host, Abby Morrison. Let's talk about science. Hi everyone, welcome to the show. Today we have Nidhi Rawat and Vijay Tiwari with us. Nidhi was born and brought up in India. She completed her PhD at the Indian Institute of Technology, Roorkee, after which she joined Kansas State University for her postdoctoral training. Currently, she is an associate professor at the University of Maryland working on disease resistance and susceptibility in crop plants. Vijay was born and brought up in India. He went to the Indian Institute of Technology, Roorkee for his PhD and then joined Oregon and Kansas State Universities for postdoctoral training. Currently, he is an associate professor at the University of Maryland working on small grain breeding and genetics. Welcome to the show. How are you both doing today?
Thank you.
Yeah.
You're doing good.
Good. Wonderful. So today we are talking about speed breeding and Fusarium head blights. Hopefully I'm pronouncing that right. I'm sure you'll correct me if I'm wrong. So to get us started, I'm hoping you can just tell us a little about this plant disease and kind of what it looks like, what plants it can affect, and just kind of the scope of the damages that it can cause.
Sure. Fusarium head blight is a fungal disease of wheat and barley. It is a big concern for growers. So what this disease does to the plants is that not only it shrivels the grains, it reduces the yield consequently, but also the fungus produces a toxin, deadly mycotoxin, which is called deoxynivalenol, and that is regulated by FDA. So farmers, if their grain is contaminated with mycotoxin DON, cannot sell their grain in market because it's not safe for consumption. Now, it's a major problem for farmers, as I mentioned before, and they use chemical fungicides to control it. But as you know, chemical fungicides have their own pluses and minuses. So a lot of times that leads to fungicide resistance emergence in pathogens. Plus also, it's not very economical for the farmers to spray it year after year. They are costly chemicals, and it's not environmentally friendly either. So genetic resistance is one of the most sustainable, economical, and environmentally friendly approach to manage this disease. So we have been fortunate that we are working together. I am a pathologist, so I work with the disease aspect of this plant pathogen and crop. And then Vijay is a wheat breeder for the state of Maryland. So we do the discoveries and then he can implement those in varieties. So we discover genes and then he can readily deploy them in the crop varieties.
So as Nidhi was mentioning that it basically creates a lot of problem for the growers and farmers for wheat and barley. The problem is that if FDA has put a limit, if a grain has produced as higher than 1 ppm of this toxin that was produces, then it cannot be used for the human consumption. So that means produce can be lost. And we have seen in Maryland some of the severe incidences of a Fusarium head blight, where growers have to come back from the mills because they cannot sell their produce. So this is bigger problem, much bigger problem, one of the growing problem with changing climate. The diseases are going to take more global acreages. So it's a global concern, not just for us in US, but across the globe. And I think just want to add up what Nidhi was saying, that fungicides have their own limitations. And if you look at how much fossil fuel is being used in developing those fungicides and kind of energy has been used and kind of bad influence they bring to the environment, it's create a much better idea to look at the sustainable part of the agriculture where plants have their own genes so that they can find these pests and pathogens like Fusarium head blight.
Sure. So obviously, sustained fungicide use is not the solution. You've mentioned that, the the genetics of resistance is kind of the long-term solution. But that also has a few drawbacks that can come up. And so you are focusing your research a lot on this idea of speed breeding. So I'm hoping you can kind of introduce our listeners to that as just kind of like a concept, how it works, why it's important to use that over the regular breeding efforts, and maybe some of the drawbacks you were hoping to address.
Okay. So I will talk from the wheat's perspective. I think Lee Hickey and his team, they came out with this approach that by applying more light and unique temperature, you can basically cut down the plant's production cycle, right? So that is one aspect. But if we come to the wheat, if you're looking at the traditional cultivar development cycle, it takes about 8 to 10 years from the first cross when a breeder makes the cross and until the variety comes out in the field in the grower's hand. That's pretty long time for cultivar development cycle. And so you are basically doing breeding for one specific trait or one particular disease. By the time that variety has come out, you will see that some other challenges has come out, right? So that's very long cycle. So idea is that can we basically work around this? And there are some approaches. For example, double haploids provide some solution to that, but all the program cannot use it because very costly approach. So speed breeding provides much easier solution. If you're working with a spring wheat, then you can take 6 generation in 1 year. That means you can go and start testing wheat in the large scale in 2 to 3 years timeline, right? That means in fifth year you can have a variety ready to release. So that means you are cutting down that cycle to half, or maybe that variety development cycle to the 60% of the initial pipeline. So that's why it's pretty huge. As when you say that like sustainable agriculture, sustainable production of the plant has its own challenges. So I think one of the biggest challenges finding right gene. And second is that if you find the right gene, then how much time it takes to put that gene into the cultivar. So speed breeding is one of the tools that cut down that developmental cycle. So that everybody has a win-win scenario.
Sure.
And talking about FHB research and how we are using our research in deploying and finding useful genes and then implementing them in a very timely manner. So Vijay's program has adopted speed breeding to develop his varieties quicker. And we just wanted to see if we can combine that with FHB evaluation of the germplasm that is there. Because now generation time is not that big of a challenge, but the challenge is how quickly we can phenotype our germplasm. So the idea for this research was to check if we can expedite also the FHB evaluation of whatever material we are generating quicker. So yeah, we'll talk more about the research that we did and how it led us to speeding up the overall generation and deployment of useful genes for fighting FHB in our
Sure.
research.
Sure. That's a great overview of this valuable technique. So you had then 3 specific questions you're hoping to tackle within this research project. I know we'll get kind of more into your setup for how to answer those, but let's start off with the questions. What were you trying to learn?
Yeah, so as Vijay was mentioning, we can cut down with speed breeding the overall generation time of wheat. So we wanted to see if we can use those conditions to speed up our FHB evaluation. So we did systematic experiments. So one of the questions that we had was, can we compare FHB severity under normal conditions versus speed breeding conditions in different genetic backgrounds, which are subtly different from one another in the resistance levels to FHB? The second question that we were trying to address is how much time we can save if we do all these experimentation with speed breeding conditions versus their normal conditions. And then third was, as I was mentioning before, we are also concerned with mycotoxin DON. So we also wanted to see if that speed breeding condition also provides us a true assessment of the DON levels that accumulate at the end of the speed breeding cycle. So these 3 were our main questions.
Sure. So I think before we talk about the specific approaches to these 3 questions, I think it's helpful to get an overview of how speed breeding works on kind of a high level. I know we've talked before on the show about Norman Borlaug's work where he was kind of using different locations, but there's also lots of different ways that you can kind of approach it. So I'm hoping you can discuss how in this research you were approaching the setup.
So this work was done under our supervision, but we were very lucky to have a very bright and enthusiastic and motivated PhD student, Bhavit, who is the first author of this paper as well. He conducted the experiments. What we did was we picked 6 different wheat genotypes, which were differing on their resistance levels. So some varieties were resistant, some varieties were very susceptible, some were moderately resistant, some were moderately susceptible, and we had a range. And what we did was we exposed them, we divided them into 2 sets. All 6 varieties were going in speed breeding settings, and then the 6 varieties were again growing in normal breeding settings in replicated experiments. And then we challenged those plants with Fusarium graminearum pathogen. And we recorded the disease progress and also evaluated DON content of both the sets of all the replicated trials with different genotypes at different time points, so 14 days and 21 days, and then at maturity. And yeah, so that's how we conducted this experiment.
Yeah. So actually, the idea behind it is that can we force the plants to get into the flowering early? So again, these are the experiments that we have conducted in the greenhouse conditions. Usually in the field, you cannot have those variables under control, right? So you have a rain and everything is gone. You have dry weather. So we wanted to make sure that we try to mimic whatever can be best condition to provide a faster generation. So we tweaked the light conditions. So we basically made sure that plants, usually you are looking at 16-hour versus 8 hours of light versus dark period when you grow the plants in the greenhouses. We fold the plant with 22 hours of dark light and 2 hours of dark. So they are staying in the light for 22 hours at elevated temperature, right? So you also increase the temperature from 3 to 4 degrees. And we made sure that we are not watering plant on the top because it's also, you're not creating a variation in terms of entropy and stuff. So everything was consistent, what every plant was giving. So that provides us an opportunity to look at those fine scale differences. And then I think Nidhi's group was pretty robust in finding out what are the best conditions for scab, right? And which are also kind of overlapping with higher temperature and more moist conditions. So it worked out pretty well. We see that under speed breeding condition, we can cut down the testing time for scab.
So my question here is related to how you're making your measurements on a crop's progress, because basically you're trying to make a plant like grow up faster in a sort of way, right? And so my question is, when you're doing your assessments of what you're measuring, are you going by, we're going to measure at this plant's like development stage regardless of how many days it is? Or is it just, I know Vijay, you mentioned, we do it at these specific dates because it feels like you're trying to measure like how quickly they're growing. So is it just by date regardless of where they are? I mean, maybe that's the whole point is to see if they're farther. So if you could just explain a little bit about how you kind of, I don't want to say sync up your data because that's not really what you're doing, but like how that measurement process works.
So that's a very good question, Abhi. Yes, you're right that things don't progress at the same level in nature, and that's the beauty of nature. But one thing with having controlled light conditions, and especially having those enhanced light periods, photo periods, is that everything happens very rapidly for the plant. So even if there is a difference in normal breeding conditions of like 2 weeks of difference in flowering times, in speed breeding, that difference will also be condensed into like 2 or 3 days. So that really homogenizes all the dates, first of all. But secondly, we do these inoculations and these various measurements on the specific stages of the plant itself. So it's not according to a calendar date that we'll record this data on this date. It's how the plant is progressing. We'll target the same stage of same plants and we'll record data. We record it every day, although it doesn't make sense when we are reporting it in the paper. So we report only the significant stages of differences that we see. So for example, in this paper, you'll see we have reported data on 14 days as well as 21 days as well as 7 days. But the significance is for 14 days versus 21 days. So that's where we focus for the paper's sake. But we keep our plants under a daily observation.
Sure.
Yeah, actually, Fusarium head blight is the disease of the wheat spike, right? So what we do is basically we inoculate the spike at the right stage, and basically we try to capture exactly same stage in all the testing samples. And then when you inoculate it, you take the data exactly after 7 days, 14 days, or 20 days interval. So you are basically homogenizing the whole process to have a nice sync. So it's a robust but a very nice process.
Yeah, and also specifically, if you ask about the specifics of when was the inoculation done, for example, so in field condition, the plant is attacked by this fungus when it is flowering. So that's when also we do phenotyping in the greenhouse when the plant is starting to flower at anthesis or pre-anthesis stage.
Awesome. Thank you for clarifying. So that's an interesting kind of mix of, wow, there's a lot of calculations going on there. So shout out to your student for taking a lot of the work on to do that. So then what were you specifically testing to get kind of your 3 questions answered?
Yeah, so to see how our hypothesis will work, we inoculated the plants at pre-anthesis stage. So just before their anthers will come out, we'll inoculate them with a definite amount of spores, which is 1 into 10 to the power 5 per ml. So we'll calculate the density of spores in the solution, and then we'll put 10 microliters of that in the flower of wheat at 2 points. And then we'll cover that with the moisture saturated bags for 72 hours, and then we'll come back to the plants. We'll remove them after 72 hours, and then we'll come back to the plant every day and see how the plants are doing. And so what we saw was that the plants which were resistant even in speed breeding conditions, or like what it was in normal conditions. Same way, the dawn levels were same whether it would be speed breeding or normal conditions. The only difference was we could see the differences much earlier in the speed breeding conditions versus our normal conditions. And another great observation was that I mentioned in the beginning that we took varieties which are subtly different in their resistant levels. We could record those subtle differences even with our speed breeding conditions. Initially, we didn't know actually how well it will respond. And when we correlated all the data, it was a pleasant surprise to see that we got above 90% correlation between our values, even though we could do everything faster.
So actually, when some of the researchers established the speed breeding protocol in bread wheat, they're talking about almost 2 months for one generation, right? So, but if you look at the Fusarium phenotyping protocol, you are taking the data after flowering when you inoculated the wheat flower, and you are taking the data 21 days after, right? So you got to have that long wait after plant flower. Whereas if you're not doing that, after 15 to 20 days, you are done with that generation, right? So one of the goal was that if we are going for spring wheat or winter wheat, how can we cut down that evaluation time? Can we take that data in 10 to 12 or 14 days to cut down those 10, 15 days? Because if you're looking at 10, 15 days in a cycle, that's not a big difference. But if you're looking at 6 times in a year, that is huge time. You are saving 2, 3 months. So first goal was that can we cut that time? Second goal was that even if you are cutting that time in the evaluation and bringing it fast in at a faster pace, can we still be have that kind of high confidence in calling the disease susceptibility or resistance, right? So disease spread and dawn content. So if you combine both the stuff, I think the third immediate answer that we are seeing is that if we have these two things checked up, then our crop development pipeline is going to be faster because we have a quick approach to test the lines for scab resistance. That means we can have another additional trait added to our pipeline that can help us in developing much better cultivars in shorter time.
Hi, everyone. I hope you're enjoying the show. Interested in learning more? Nidhi and Vijay's paper, Evaluation of Speed Breeding Conditions for Accelerating Fusarium Head Blight and Deoxynivalenol Screening in Wheat, published in Crop Science, is always freely available. If you are a certified crop advisor or certified professional soil scientist, you can take a quiz for continuing education units for this episode, which can be found in our show notes or on certifiedcropadvisor.org. Thank you for being our sponsor. Let's get back to the show. I'm finding that because I am who I am and don't come from a science or agricultural background, can we lay like a couple basics on like how from like planting the seed to we've confirmed what we need to confirm for this cultivar, like how long was that in your control? And then like, yeah, let's start with that. I don't really know the developmental stages of wheat that much, so I'm just curious.
You want me to answer, Nidhi, or you want?
Yeah, you can answer it.
So I think in US we grow winter wheat, and in some state we also have a spring wheat, right? So this protocol goes differently for winter wheat and spring wheat, right? So paper that we are talking about works on spring wheat. So as I said that there's established protocol that if you do everything right, you can have a full generation from planting to harvesting in 60 to 65 days. That's pretty fast, right? So, but in that case, you are not moving a lot of plants' energy in establishing itself, right? So you put plants in a pot. If you're working with the spring wheat, in winter wheat, it's a totally different strategy because they require core treatment or vernalization, right? So if you're working with a spring wheat, we put them in the pot and then let them acclimatize for a couple of days. And when they germinate, you just get them that environment of higher temperature and higher light. And then you keep them at the same place, just not watering on the top so that the energy is not consumed in maintaining the temperature on their surface. So that's how the plant has just one goal to keep growing because they're getting enough nutrients. So they just do what they can do the best to grow.
Sure. That's super helpful because I think when you think about, 65 days and then some of the time frames you've been throwing out as far as how much you could really shorten that, I think that really clarifies like how much of an impact that has. Speaking of, I would love to hear more about your results of what you found for your answers for your questions.
Yeah. So we found that we can actually use speed breeding to clearly determine what is resistant and what is not. And even the minor differences between moderately resistant to moderately susceptible, we could see those differences, statistically significant differences in all the genotypes in all the reps. And the correlation values that we got between the severity and dawn in speed breeding versus normal growing conditions were upward of 0.9. So they were great. And what we found that using speed breeding conditions just for the data recording time, we could save at least 1 week. But if you are starting with your plants in speed breeding conditions in the beginning, and then you also do FHB phenotyping in speed breeding condition, overall it saves you around 1 month. And when you're talking, as Vijay was saying, we can take 6 generations in 1 year. And if you're saving 1 month just in one generation, then you can calculate how much saving of time that is. So yeah, that was we found with our research.
That's awesome. That's incredible to be able to like have the time is wild. Oh man, I so love having anything to do with plant breeding on this show because I just think it's like the funnest thing. But I'm always so happy when they're able to cut down the time because that's like hands down is always kind of the big issue to hold back progress. So this is wonderful news. So it sounds like you were mostly just kind of testing cultivars that you already knew had certain levels, but haven't quite maybe progressed into now like doing crosses in this. So I would love to hear like about that if you're doing that, as well as any other future research that might be on the horizon for this.
Yes, definitely. So first of all, we are using speed breeding for our variety development program. Vijay is using that constantly. So because of that, he joined the University of Maryland as a assistant professor, as a plant breeder, as a wheat breeder in 2020. And just in a matter of 5 years, so he is releasing some varieties this year. So that is actually what we are saying in the paper. We are actually showing it with our breeding program itself. So that is one success story. We plan to use the FHB evaluation protocol under speed breeding conditions also for incorporating in our breeding program now. The other thing that we want to do is that, as Vijay was mentioning, this all study, because we wanted everything regulated and constant, we did all these studies with spring wheat varieties with known levels of genetic resistance. But now in Maryland, especially, we grow winter wheat varieties. So we are trying to replicate the same setups in winter wheat evaluation. And then also we used a very aggressive isolate for this study, which we have used in our published research as well previously. But actually, fortunately, in field conditions, we don't have that aggressive races. So we also want to see how these conditions will respond to the actual strains that we collect in the field. And so next step will be to incorporate everything what we have learned so far in our breeding program and make the gains of all the research that we are doing reach the farmers faster.
So I think I would like to add on what Nidhi was saying, that I'm a breeder, so I am basically utilizing this wonderful collaboration that we have with Nidhi. She basically is, and parallelly we are evaluating lot of lines with Nidhi for our scab resistance, and we have been putting lot of FHB resistant gene in our breeding pipeline. So I think it would have not been possible if speed breeding was not the case, right? Because we are turning the tide very quickly by doing some marker selection in house and selecting the lines which we think may be resistant. But before you go to the field, you got to make sure that those lines are actually resistant. And before we harvest lines in the greenhouse and going to the field, we have very small window, right? So Nidhi's program basically get in and test those lines in the greenhouse, and her program tells us that, okay, don't waste your energy on those lines because we don't feel that you see any resistance out of it. So it's very collaborative approach where phenotyping and genotyping and evaluations are going parallelly with the cultivar development pipeline. Second thing is that, as Nidhi was mentioning, that this paper is focused on the spring wheat, and we have established a very robust pipeline for speed breeding in the winter wheat germplasm. I'm so glad that different programs in the US are also now adopting that. But still, we are not at the pace where we could work with the spring wheat. In spring wheat, you can get one generation in 2 months. But in winter wheat, you have to vernalize the plant. So right now in the future research, we are trying different options to fool the plant so that they can cut down their vernalization requirement. That's 6 weeks of core treatment they require. So we are thinking if we can cut down to 2 weeks, 3 weeks, or 4 weeks, then still we are going to save couple of weeks. We are seeing some very exciting results, but these are some more confirmation that we need to do before we can conclude this one. So we are involved in cutting this cycle further down.
Oh man, this whole episode is just like warming my heart. That's like such a nerd. I love it. It makes me so happy because I didn't realize that you like already had some cultivars that were like making it because of this research in half the time. That's so cool. But I'm gonna try and control my fangirlishness about this a little bit and move us on. We have 3 questions left for you. So the first one is, if listeners want to learn more about this research, where can they go?
Yeah, so USDA has established a um, initiative that is called US Wheat and Barley Scab Initiative, which is dedicated to do research to solve this problem of FHB in wheat and barley. So it supports research in public university as well as USDA federal labs, research institutes from all directions so that this challenge can be mitigated. And I will provide link to the SCAB initiative website. And not only our research on genetic resistance is a mandate, but also your scab initiative works with farmers on how to develop best management practices, be in terms of crop rotation, cultural management practices, or even fungicide usage, or even if there are new chemistries that are being developed, they are tested under the umbrella of scab initiative. So I think scab US wheat barley scab initiative should be continued to support it and to be supported so that farmers can be benefited and can actually be helped with mitigating this FHB challenge.
Yeah, I think I would like to add what Nidhi was saying, that wheat barley scab initiative is fundamentally very important platform for researchers, growers, and wheat and barley stakeholder across the globe. That basically is a very essential platform for developing FHB resistance, which is going to be much bigger problem in years to come. We see incidences in US are going to go increase with higher temperature environment and more moist climate, and it is going to go to Africa, where nobody has heard about Fusarium head blights getting one of the problem there. So it is going to be a global concern, and wheat barley scab initiative is playing such an important role in controlling this devastating disease. So I hope support will continue toward that effort. But for getting more information about this research, listeners can contact us directly to our email IDs. My email ID is vktiwari@umd.edu. I have a web page at University of Maryland. They can look at the information there. I will be very happy to talk to them over a phone as well. Our website has listed our phone numbers. So there are multiple sources to get the information, as well as some of the publication that we have been fortunate enough to put out in the public space. So those are the places listener can get more information about our work.
Perfect. Yeah, we'll be sure to include links to those assets in the show notes so listeners can check that out. Second question is, if people then want to take the next step and kind of get involved in this research, whether that's as a researcher or a farmer, what can they do?
As I said, that we are just a call or email away. We would love to talk to them about how we can get to work together. Again, 2 or 4 hands are not enough to solve this big problem. So that's why more hands, more people are needed to basically carry on this initiative. And I think there are many researchers are basically already joining, moving towards employing speed breeding in their breeding pipeline. So I think it looks very promising and very exciting as of now.
Awesome. Yeah, that's definitely lend a hand if that's what you're into out there. We hope we'll find some connections for you. So then, final question is, what is one fun fact that listeners wouldn't know about you if all they had was your research?
I just love talking. I just, I just love talking about anything that relates with wheat. So uh, even my chair, he always said that you talk to Vijay in 30 seconds, he will just bring wheat and he can never stop. So, so that is one thing, and I love listening some music and playing with the kids. That's it.
Yeah. And even our kids know wheat genetics and wheat genome and Fusarium head blight, the disease. Yeah.
Gotta start them early.
Yeah.
And they are 8 to 10 years old. So
Aw. Wow. Yeah, that is starting early. That's wonderful. That's wonderful. It's what we need. Well, perfect. This has been so wonderful to have you on sharing your awesome research that is going to really help tighten those timelines and help us find the solutions we need faster. So, yeah, thank you so much for being on the show and for taking the time.
thank you so much.
Thank you for having us. As Vijay was saying, he just loves talking about wheat. So you gave him that perfect chance. Thank you.
You're welcome. Hi, everyone, and welcome to our Student Spotlight, where we highlight the work of graduate and undergraduate society student members. Today, we'll be talking to Saptarshi. Welcome to the show. Can you start us off by introducing yourself and where you are studying?
Hi, Abhi. I'm Saptarshi Mondal from Calcutta, India. And presently, I'm pursuing my PhD at the Institute of Plant Breeding, Genetics and Genomics, University of Georgia, Griffin campus. So my prior degrees include an MS in plant breeding and genetics from Punjab Agricultural University, which is situated in Ludhiana, India, and bachelor's in agriculture from Vishwabharati Shantiniketan.
Awesome. And what are you currently researching?
Well, my doctoral research is being conducted under the supervision of Dr. David Jesperson, and he asked me to fight against salt. So I'm now putting my efforts into understanding the genetics of salt tolerance in zoysia grass, which is a popular turf grass in the United States, also in other countries like Japan, that is also very popular, and finger millet, which is an underutilized but highly nutritious food crop.
Great. And if you could have your dream research project, what would that look like?
So, talking about my research interest, I always wanted to be a potential scientist in the field of crop improvement to contribute my best efforts towards the welfare of agriculture. So, crop improvement through conventional breeding, but integrated with modern technologies like high throughput phenotyping, genomic modeling, also genomic editing techniques. Those are my research fascinations. I want to do a kind of a multidisciplinary research where I can put my effort in all of these techniques in together. Yeah.
Awesome. Well, if you'd like to get in touch with Saptarshi about his work, we'll have his contact information in our show notes. Thank you so much for being on the show today, and best wishes on your future studies and career. Thank you.
Thank you so much. Yeah, I appreciate your effort. Thank you.
Thank you for listening to Field Lab Earth. More information can be found in the description below. Thank you.